Okay, so in this topic, we are going to actually learn about, firstly, explain what is meant by an antigen, and then state the differences between self-antigens and non-self-antigen. Secondly, you're going to learn how to describe the sequence of events in a primary immune response, okay, with reference to macrophages, B-lymphocytes, including plasma cells, T-lymphocytes, which include T helper cell and T killer cells. So basically, these are all that we're going to cover for this lesson here. So firstly, we're going to learn about immune response, right? So therefore, you need to know the definition of immune response. So immune response is the complex series of reactions. That means it involves many, many steps, right? It's a complex series of reactions of the body in response to the entry of a foreign or non-self antigen. So this is the definition, okay? So this complex of series of reactions will involve the cells of our immune system, okay? So the cells of immune system includes mainly the two types of white blood cells that we have, which are the phagocytes as well as the lymphocytes. Okay. So what kind of reactions? So there are two possible types of reaction. A, it involves specific antibodies to be secreted. So this kind of immune response is called the humoral response. Okay, humoral response. Why humoral? Because humoral stands for liquid or fluid. So it is a liquid or the fluid, which is the antibody that act on the non-self antigen. So that's why it's called humoral response. Okay, or it can end or involve the TC or TK cells being produced against the foreign antigen. So either antibodies produced against the antigen or the TC, TK cells produced against that antigen, which is non-self or foreign antigen. Okay, what is TC and TK? TC stands for T cytotoxic cell. TK stands for T killer cell. They are the same cell with the two possible type of names. Okay, same cell, two possible types of name. Okay, so this is the full definition of immune response. This is the full definitions. Okay, so for the TK or TC cell, the immune response that involves them is called the cell-mediated response. So immune response got two types. When it leads to production of antibody, it's called humoral response. When it leads to activation of TC or TK cells, it's called cell-mediated response. Why? Because over here, it is a humor, means the liquid, which is the antibody that will act on the foreign or non-self antigen. In cell-mediated response, it is the cell, which is our TK and TC cell, or TC or TK cell, that will act against our abnormal cell of our body, which carries the non-self antigen. So it is the cell that acts on the non-self antigen. This is the antibody in the form of liquid that act on the non-self antigen. So that's why humoral versus cell-mediated. Okay. So what is antigen? You learn the word antigen in chapter 4 when you learn the membrane structure. But the definition of antigen for this chapter, which is immunity, is broader. Okay, so let's look at this definition of antigen. Antigen in this immunity chapter, it means any large molecules which is recognized by the body as foreign or non-self, and therefore it will stimulate our immune response. So as long as this antigen is recognized as foreign or non-self, it will lead to a stimulation of our immune response. So what are the possible types of the large molecules? It can be anything here: protein, lipid, carbohydrate, glycoprotein, bacterial toxin. So bacterial toxin, you see, Bacterial toxin is not name tag of the cell, because in chapter 4, you learn that antigen is the name tag of the cell, right? Name tag of the cell. And then for our own cell, the name tag is called the self-antigen, right? For other cells not from our body, the name tag on the cell is called the non-self-antigen, right? So, but then when it comes to immune response, as long as this molecule activate immune response, is considered as antigen. So bacterial toxin is not the name tag of the cell. It is actually chemical that are being secreted by bacteria, and it is toxic to our cell. So it is also considered as non-self antigen, because these molecules are foreign in our body. It can activate the immune response. So therefore, they are also considered as non-self antigen. So in this case, antigen does not have to necessarily be the name tag of the cell. You see that? As long as it's foreign, and then our body immune system recognizes it as foreign or non-self, you start to launch immune response against it. Okay. Another example is pollen grains, right? Coming into our body. Some people will be sensitive to it, and it will become allergy to it. So for your information, allergy is a type of immune response. When you say allergy, allergy is actually a type of immune response, okay, whereby our body launched the immune response against a harmless foreign molecule or particle. So because it's foreign, it's considered as non-self antigen. So that's why we launch a immune response against it, and that immune response is considered as allergy. Okay, allergy. That's why there's inflammation in the part that's allergy. You feel hot because more blood flows to that area. You know, inflammation, right? The capillary becomes dilated, so more blood flows there, becomes hotter, and then the capillary becomes leaky. Right, tissue, blood plasma goes into the tissue, and then white blood cells migrate into the tissue, so the tissue becomes swollen. Okay. So the specificity of the antigen. So specificity means refer to the specific shape of the antigen, because different antigen is made up of protein, have got different amino acid sequence, different carbohydrate have got different structure as well. Okay. It allows the responses that are customized to specific pathogens. So that means to say that in our immune response here, the presence of the antigen will activate immune response against that foreign antigen. So it's very specific. Okay, it's very specific. So it's also considered specific immune response. Now, what about antibody? Antibody is a glycoprotein. So it's made up of glyco means carbohydrate. Okay. Protein means protein. So it's a molecule made up of carbohydrate and protein, which is considered as immunoglobulin. That means globular protein involved in immune response. So they are actually made up by plasma cells, which are derived from the B-lymphocytes, and they are secreted in response to the foreign or non-self antigen. Therefore, they are specifically targeted towards that particular foreign or non-self antigen only. Why is it specific? Because the antibody got a variable region which is complementary to the shape of the specific antigen. So this is the definition of antibody. We will look at antibody structure and function in a separate lesson. So here you only look at the definition, just in case you want to know what is antibody. Okay. So what about self and non-self? Self means any substances, including the cell, that are being produced by the body. So the immune system will not recognize as foreign, so they will not launch they will not launch an immune response towards it. Okay. Now, but if this if it's introduced to another body, then they become non-self. So something produced from in our body is called self, but when it goes into the body of another person, it will be considered as non-self. Okay. Non-self, like non-self antigen, are any substances or cell that is recognized by immune system as being foreign. Okay. So therefore, it will stimulate our immune response. Okay. So now we're going to move into study the sequence of events that happen during an immune response. So it involves 3 stages. Stage number 1 is called a non-specific immune response, where our phagocytic white blood cells will go and engulf many foreign organisms by phagocytosis or endocytosis. So as long as the organism is foreign or the cell is foreign, right, our phagocytic white blood cells will go and engulf them by phagocytosis. So we've got 2 types of white blood cells that you have learned that will do this task, which is neutrophil as well as macrophages. So both of them will also phagocytose the foreign cells in our body. But the difference is that neutrophils, after they phagocytose, they will die. So they're short-lived. So they will die and they become part of the pus in the region where infection occurs, right? So they will die. Whereas macrophages, they are long-lived, they will not die. They're supposed to be moved inside our body or circulate in our body, and their main function is to present the non-self antigen in order to launch the specific immune response. Okay, so first step is non-specific. Your phagocytes will not care what are the cells. Is it a HIV? Is it a Vibrio cholerae? Is it a Plasmodium falciparum? It will not care. As long as foreign, it will go and phagocytose. So this is non-specific. So after phagocytosis, then the specific immune response will be initiated by our macrophages. That's why they're supposed to be long-lived. Okay, because they're supposed to go around the body and inform our immune system that, hey, please produce more soldiers to remove this particular like HIV or this particular Vibrio cholerae or Plasmodium falciparum and so on. Okay, so therefore it will launch a specific immune response. But the immune response will start from here first. Okay, it will start from here first. So that's why the first one you're supposed to learn is macrophages, right? The first step involves macrophages. Now, in specific immune response, it involves specific clones of B cells and T cells. So B and T lymphocytes are also called B cells and T cells. Why clone? Because you know the meaning of the word clone, okay? So if you say a clone of cell, it's actually a group of cells which are a group of cells which are genetically identical because they are produced by repeated mitotic division from an original parental cell. So that's the meaning of a clone of cells. So the clone of cells, they are all daughter cells that are produced by repeated mitotic division from an original single parent cell. So that's why from 1 becomes 2, becomes 4, 8, 16, 32, and so on. To become a clone of cells genetically identical. Okay, so clone of B and T lymphocytes can then be activated to recognize that specific antigen only. Okay, that specific antigen only. Okay. So then when they recognize it, then they will launch a specific immune response just towards that specific antigen. So that's why they only target a HIV virus. Okay, they only target HIV virus. They don't target, you got Vibrio cholerae, they don't care. Okay, Plasmodium falciparum, don't care. Right, this is called specific immune response. They only target to that specific foreign antigen that activates them. Okay. So it involved the primary phase and secondary phase, where primary phase is actually all of you have learned it as the primary immune response. Okay, primary immune response. And then the secondary phase is considered as your secondary immune response. Okay, so what is primary and secondary? Primary is when your body is exposed to that pathogen for the first time. For the first time, then your immune response is called primary response. But after exposure for the first time, subsequently, let's say 2 years later, you are being exposed again to the same type of pathogen. Okay, then your body will launch immune response. The immune response is called secondary phase already, or secondary immune response. So that's the difference between primary and secondary. Okay. Okay, so now this is a definition of specific immune response. It's very wordy. I will go through with you, but I prefer to use a diagram to guide you so you understand better. But we will go through this definition first. Then when I use diagram to describe, you will understand fully. Now you might not understand yet, okay, but it doesn't matter, this definition. So what is a primary phase of the immune response or primary immune response? Specific clones of B cells, okay, divide and differentiate to form antibody-secreting plasma cells. And then specific clones of T helper cells or T killer cells, also known as T cytotoxic cells, divide and differentiate to form active T helper cells or active TK cells and memory cells. So this is what happened to them during primary immune response. Okay, so the products, they are specific to the antigen and that will contribute to the destruction of the specific antigen and preventing its activity. So what are the products? After you will see the products. Okay, the antibodies is antibody is the product. Activated T helper cell and activated TK TC cell are the products. So you look at that in detail in the diagram. Okay. Now what about secondary phase? For secondary phase is when the memory cells will act because they remember the pathogen that entered the body 2 years ago. Right now, the same species of pathogen enter the body, so they can recognize it because it's called the memory cells. So they retain the capability to secrete antibodies or to act as TH or TK cells as soon as the specific antigen is detected again. So let's look at the whole story. All right. Now, this is the main diagram that you want to use to learn about immune response, and this will be your main notes as well for the primary immune response. Okay, start from the step number 1. So we are going to go through the sequence of events or sequence of reactions that occur during a primary immune response, starting from number 1. So this is an example of a bacterium with antigens on its surface. So it happens to enter our body through an open wound or open cut here. So from external environment, it goes into internal environment. Okay, so can you tell me the examples of internal environment in our body? Samir? So your internal environment is tissue fluid, blood plasma, and lymphatic fluid that bathe our cells in our body. So once the bacteria enter our body, step number 1 of the immune response is the phagocytes will start to phagocytose it. So we got the neutrophil and macrophage. So in this case here, we're going to talk about the macrophage, okay, because it involves macrophage to activate the entire immune response. So macrophages will phagocytose the bacteria, but it will not fully digest the bacteria like neutrophil, because its main purpose is actually to cut the bacteria into small pieces and then take all these small pieces of the bacteria which contain the antigen and present it on the surface of the macrophage itself. So it can be presenting directly on the cell surface membrane here, or it can be, or the antigen of the bacteria can be held by a particular receptor. So say, for example, this is a receptor here, and then it'll be holding the antigen of the, hang on, this one cannot see clearly. So use blue color, green color. Okay, so it will be holding the antigen of the bacteria, triangular shape. Okay, so either hold directly on the membrane or hold on a receptor for the antigen. So this step number 1 is called antigen presentation. So that's why macrophage is long-lived, because they're supposed to move around the body to present the antigen that of the bacteria that is phagocytosed. So at this time now, macrophage is called the antigen presenting cell, APC cell, antigen presenting cell. Okay, so after that, the next step will be called the clonal selection. Clonal selection means the clone of B cells and T helper cells will be selected. So what do you mean by that? You see here, I've got B1, B2, B3 cells, right? Okay. Can you tell me what are the differences between B1, B2, and B3 cells? So you can see that B1, B2, B3 are different because of this molecule on the surface of the cell, right? So this molecule here, right? This one here, and also this one here. Because this is U-shape, this is V-shape, and this is actually straight like this. Okay, this is V-shape here, right? And this is U-shape here. So they're all different because of the receptor molecule on the membrane that's different. So this receptor molecule is actually called the antibody receptors. So they are called the antibody receptors. Okay, I tell you why they are called antibody receptors later on. So that's why there are 3 different types of B cells here. Okay, so what happened in our body is that our body, when we produce the B cells, we will produce a clone of B1 cells. So we got a clone of them. That means you got a group of B1 cells. Okay, we got a group of B2 cells. We got a group of B3 cells. So their clone means they're all genetically identical. So they exist in clone. Okay, but we only show three different B cells here. Right, and also you look at the T helper cell. This is TH, TH1, TH2, TH3. You can see not TH1, TH2, TH3. So these T helper cells are also different in terms of the receptor, T cell receptor. Okay, the receptor on this cell is called the T cell receptor, which is versus this is antibody receptor on B cells. Okay. So where's my T cell receptor? The green color one. You see, I got a red color and green color. The green color ones are T cell receptors. The shape of the binding site for these 3 are different. Okay, not easy to see, but it's different. You can see that this is square here, right? The shape of this binding site here is square, right? So this is square here. Whereas the shape of binding site, this one looks more like cannot see. You all can see, you know. One of them is U-shape and one of them is V-shape. So I think this one is a V-shape. So it's V-shape here, and then the other one is a U-shape. So the binding site is U-shape here. So all the 3 receptor has got different shape of binding site. So therefore, different T helper cell. So T helper 1, T helper 2, and T helper 3. So you've got 3 different clones of T helper cells. Okay. So what happened in my clonal selection is that the clone of B cells with the receptor that's able to bind to the presented bacterial antigen, okay, it will be selected. So in this case here, this B2 cells, the clone of B2 cells will be selected. And then in this case here, the clone of TH2 cells will be selected. Now the detail you don't have to know, like what is CD4, what is MHC protein, no need to know. No need to know. Okay, just know it's receptor, finish. Okay, so now, so this is called clonal selection. All the T helper cells and the B cells with a receptor that is able to bind to the antigen of the bacteria presented on the surface of APC will be selected. So this is called clonal selection. This is a name given to this process. Please remember this name, clonal selection. So after clonal selection is clonal expansion. Okay, clonal expansion. So when you see the word expansion, means increase in size or increase in number, right? So we're going to increase in the number of the cells that are being selected. So how to increase in number? Step 1, the selected T helper cell will divide by mitosis first. Okay, they divide by mitosis. After that, they will differentiate into 2 types of cells. So this is called clonal expansion. The cell will divide by mitosis and then differentiate into 2 different types of cells. Number 1 is called the active T helper cell, and number 2 is called the memory cells. The memory cells. So you can remember the antigen already entered the body. Okay, so got active T helper cell and T helper memory cells. Okay, now after T helper cell has undergone clonal expansion, an active T helper cell produced already. Then this active T helper cell, now I've told you before, all the lymphocytes, be it B lymphocytes or T lymphocytes, they function by secreting chemicals. So the active T helper cell function by secreting chemical called cytokine, which is also called lymphokine. It's the same thing. Lymphokine is the chemical secreted by lymphocyte. The T cells are lymphocyte. Cytokine is a chemical secreted by cyto means cell. So cytokine is a more general term. Lymphokine is a more specific term. They are the same thing. Okay. So active T helper cell will secrete the cytokine. Now this cytokine will then stimulate clonal expansion of the selected B cell. So this B2 cell already selected, but it will not undergo clonal expansion until it is stimulated by the cytokine secreted by active T helper cell. Then only it will undergo clonal expansion, means it will first divide by mitosis. Okay, so divide by mitosis. And after that, it will differentiate into two types of cell. The active cell is called the plasma cells, and the remainder cells are called the memory cells. Okay. So you ask me, teacher, why T helper cell must undergo clonal expansion before B cells can? Then you have to know the role of T helper cell. T helper cell is like the manager, the director, okay, of our immune system. So this is the big boss. So the big boss have to give instruction. Then only the subordinate will go and do the work. Subordinate, okay, will go and do the work. So therefore, T helper cell is actually the manager of our immune system. So the rest of the cells have to wait for the instruction from the manager before it will do anything. So even though B2 cells already selected already, but it will not do anything until the manager, the T helper cell, secrete cytokine to inform it, okay, now you can divide. So then the B cells will start to divide, because now I've got a lot of these antigens, so you have to divide. So the function of cytokine is not only to activate the selected B cells to divide, it can also activate macrophages to divide. It can also activate selected TC cell to divide, because all these cells are cells of immune system. So therefore, T helper cell is the boss, the manager of the immune system. Okay. So what happened next? So after all this process up to here already, now once plasma cells are produced, the plasma cell will secrete antibody. So can you see the antibody molecules looks similar to the shape of the receptor? Right? This is the antibody molecule, right? It looks similar to the shape of the antibody receptor. So actually, the receptor molecule structure is similar to the structure of antibody. That's why it is being called the antibody receptor, because it looks like antibody. So you know that B2 cells, once activated, secrete antibody will look like the receptor. B3 cells secrete antibody, it will look like this. B1 cells antibody will look like this. Okay. So after antibody being secreted, what's next? The antibody will then circulate in the body, because it's secreted into the tissue fluid, into the blood, into the lymph, so continue to circulate between these three fluids in our body. And then when it encounters the same type of antigen, it will be able to bind to the antigen. So it says here antibody coats the bacteria, because the bacteria contain the same type of antigen. So it will bind to the antigen due to the specificity of the antibody. So that means by complementary shape between antibody and also the antigen. So it will bind. So the moment it binds to the bacteria, it will actually coat the bacteria to be removed by phagocytes. Okay, it will code the bacteria to be removed by phagocytes. So which means to say it will actually mark the bacteria to be removed by phagocytes. And therefore, these antibodies are called the markers. Okay, because our phagocytes will contain receptor for the antibody as well. So it will promote the antibody-coated bacteria to bind to the macrophages and neutrophil to be removed by phagocytosis. So therefore, the next step is phagocytosis. And then whatever remains will be our memory cells, which include memory cells of our T cells or T cell memory cells, T memory cells, and B memory cells will remain. Okay, so this is a full description of the sequence of events that occur during primary immune response. And whatever I've shown you here is the humoral response because it involves secretion of antibody by the plasma cell. So you need to spend time to learn up the correct name and learn up the steps and learn up the name of each step and learn up the name of the cell produced, the name of chemical secreted. Okay, so we need to learn from this diagram. So after this, let's look at the structure of the B cells. Okay, not B cell, sorry, plasma cell. So plasma cell is the cell that will secrete antibody. So you know that the organelles that are present in a cell actually depends on the function of the cell. So the function of the cell is supposed to secrete a lot of antibodies, which is actually glycoprotein. You expect to find a lot of ribosomes, a lot of rough ER, a lot of mitochondria, Golgi apparatus, secretory vesicles in the cell, and the nucleus will be quite prominent. Prominent means big. Okay, why big? Because there are a lot of protein to be synthesized, so there'll be a lot of transcription. Transcription happens in the nucleus. So a lot of DNA has to be opened up so that the gene will be exposed, so the bases can be copied, okay, to form the RNA. So you can see here a lot of rough ER, and then you've got Golgi. Golgi bodies are not clearly seen here. All right, you're supposed to be able to see a lot of Golgi body, but here I don't see Golgi body. Right, it could be some other location of the cell because you only see 2D here. So certain part you can see a lot of Golgi body, and here you can see this mitochondria here. Right, all these are mitochondria. Okay. Now, just now we looked at the humoral response where the B cells are being activated in order to produce plasma cells and memory cells, and plasma cells secrete antibody. Now we're going to look at cell-mediated response where the TK cells will be activated. Okay, so in the same way, we've got an APC cell that presents the antigen. So the T helper cell and killer T cells will be activated. Clonal selection. So the step number 1 is clonal selection. All right. So clonal selection. So after antigen presentation okay, by the APC cell, it will be clonal selection. So our T helper cell and T killer cell, which contain receptor which can bind to the presented antigen, will be able to bind to the antigen and therefore undergo clonal selection, means they're selected already. Okay, so after clonal selection, number 3 is clonal expansion. Okay, so clonal expansion means the selected cell will divide and differentiate into 2 types of cell. Number 1, the active cell. Number 2, the memory cell. Of course, the active cell, if they got a specific name, you have to give them the specific name, like plasma cell. Okay, so over here, T helper cell undergo clonal selection. After that, clonal expansion to divide by mitosis and differentiate into memory cells and active T helper cell. Okay, so active T helper cell will secrete the cytokine to stimulate the selected B cells as well as a selected killer T cell or T killer cell to undergo clonal expansion because they're already selected. So undergo clonal expansion means the cell will divide by mitosis. Okay, and then it will differentiate into, so divide and differentiate into the active cell and the memory cell. So the active cell here is called the active TK cell. Okay, so active TK cell will launch an attack on what cell? Okay, TK cell or TC cell, they will only attack 3 abnormal cells in our body. 3 types of abnormal cells in our body. Number 1, infected cells. That means our body cells infected by virus, infected by bacteria, they become abnormal. Number 2, cancer cells, so also abnormal cell. Number 3, cells of transplanted organ. Okay, that means the cells of transplanted organ are considered as foreign. So that's why our T killer cell can attack them. So you can see that all these 3 cells are actually human cells, right, in our body. But they are abnormal human cells. So all abnormal human cells will be killed by TK cell. That's a T killer cell. They're supposed to kill our abnormal cell. Why we know that they're abnormal? Because all these 3 abnormal cells will present the non-self antigen on the surface of the cell. Non-self antigen. Okay, for example, infected cell, let's say infected by the virus, it will start to present the viral antigen on its surface. So suddenly there's a foreign antigen on the surface of a cell. Of course, there can be a few, right? Not only one. Okay, so it's presented. Or cancer cell will have their own antigen, which is abnormal, so it's considered as non-self antigen also. And of course, cell of transplanted organ is originally not produced by us, so the antigen is also considered as foreign. So all these 3 types of cell will be removed by T killer cell. So the active T killer cell, how does it remove the 3 types of abnormal cells in the body is step number 1. It must first move towards and bind to our abnormal cell, which has presented that non-self antigen. Okay, so it must bind to the cell with a receptor binding to the non-self antigen, same as how it binds to this presented antigen here. Okay, so after binding, what it will do over here, okay, is being enlarged in this diagram. So step number 1 is it will secrete perforin, secrete perforin, which is a protein molecule, group of protein molecules, which will punch a hole on the cell surface membrane of the targeted cell, which is the abnormal cell that it has already bound to. So you see here, they got 2 lines. Can you see these 2 lines forming a tube across the cell surface membrane? So these 2 lines are the protein from the perforin molecule. Okay, so it's secreted by our TK cell. So after it secrete and punch a hole, then it will secrete a second chemical. So the second chemical is actually number 2 here, which is made up of H2O2 and free radicals. Okay, so how is it being secreted? Very easy. You've got the secretory vesicle here, move towards the membrane, fuses with the membrane and release the content by exocytosis. And those chemicals will diffuse across the tissue fluid, move past the perforin and enters the cytoplasm of the cell and start to oxidize all the chemical molecules inside the cell. So that's how they will kill the cell. So using H2O2 and free radicals. Okay, so these are also the chemicals that are found in our lysosome, because if you remember, when you learn lysosome, there are 2 main types of chemicals in the lysosome. H2O2 and free radicals are used to kill the bacteria, Whereas you also got enzymes that are used to digest certain molecules by hydrolysis, okay, to break certain bonds in the polymer. Right, so these are the two different types of chemicals you can find in the lysosome. But in this T killer cell, it will only secrete H2O2 and free radicals because it's very effective to oxidize, okay, to oxidize all the chemicals to kill the cell. So therefore, from here, you can see that cancer cell can be removed by our cells of immune system. Unless the cells of immune system are not working properly, then the cancer cell may actually continue to grow. Okay. So then you have to think of, so what are the conditions needed in order for cells in the body to be healthy and working in an optimum condition? Probably it needs enough oxygen. It needs an optimum temperature. It needs an optimum pH. So usually the area around the cancer cells are very acidic. And it says that cancer cells thrive in an acidic environment. Why acidic? Probably there's a lot of CO2 release and the circulation is not good, so the oxygen cannot be replenished and CO2 cannot be removed from there. So then your immune system cell cannot function properly already because circulation is not good, means temperature is low. Then pH is also low, acidic. So our cells of immune system cannot function efficiently because it's not an optimum condition for them to function. So these are something that we need to take care of, right? So this is a very interesting diagram to show you that you see here, these are the two TK cells, and this is another cell in the body which is larger than TK cell, because remember, lymphocytes are the smallest white blood cells. Okay, it can be either the same size as red blood cell or slightly bigger. So they are actually, they look smaller than this cell here. So they have already attached to the cell, and probably they are punching hole and secreting the toxic free radicals into the cell to kill this abnormal cell of the body. Now, in this case here, these cells here are the red blood cells, and this is the T killer cell. So T killer cell, remember you've got two types of lymphocytes. One is same size as red blood cell, the other one is bigger. So this is a bigger lymphocyte. So already bind to a few red blood cells. That means these red blood cells are considered as foreign. Okay, so that's why they attack those cells. So it could be because of mistake in blood transfusion, right? Let's say your blood group B and a blood group A, blood from blood group A is being transfused to you. So all these red blood cells will be carrying antigen A. So of course, they will remove them because foreign antigen. Antigen A is a foreign antigen because your body red blood cells antigen B. But this antigen A, okay, non-self antigen. So what you have learned today, let's recap. You have learned the term antigen, self versus non-self antigen. Okay, you have learned about the specific immune response, okay, or the adaptive immune response where we produce antibody which are direct against a specific antigen, and we produce T killer cells that will act directly against a certain antigen, certain non-self antigen, so specific, okay? So it first involves antigen presentation. So this is the name of that step. So remember this name, you write this name is 1 mark before you elaborate. Second step is clonal selection, where the B cells and T helper cells, T killer cells, with a receptor that can recognize and bind to that foreign antigen will undergo clonal selection, followed by clonal expansion starting from T helper cell first, which is the manager. Okay, so divide and differentiate into active T helper cell and T helper memory cells. So after that, the active T helper cell will secrete cytokine, which is a chemical that will activate mitosis or cell division of other cells of immune system. So it will cause the selected B cells, as well as TK cells, as well as the macrophages to divide. Okay, so to divide by mitosis. So that means they'll undergo clonal expansion, means divide and differentiate to form active cell and memory cells. Okay, so for B cells, it will become plasma cell, and the plasma cell will secrete antibody. And antibody can be used to coat the bacteria as well as to kill the bacteria. No, sorry, it will coat the bacteria and it can also kill the bacteria. Okay, T killer cells after undergo clonal expansion becomes active T killer cells and T killer memory cells. So active T killer cells are going to kill the target cells, which are the 3 types of abnormal cells in our body. Okay, so therefore, this whole series of steps are the sequence of steps involved in the immune response in our body, which can be further divided into humoral response, which involve B cell activation in order to secrete antibody, or number 2, which involve TK cell activation. This is called cell-mediated response. Okay, because humoral response is the antibody that act against the antigen, the liquid. Okay, the soluble antibody is a liquid, act against antigen. Cell-mediated response is the T killer cell itself will bind to the cell to remove the cell. So it's called cell-mediated response. And how does it remove the cell? By secreting chemical. Number 1, perforin. Number 2, H2O2 and toxic free radicals. Okay, so your homework is to go back and revise this whole section and make sure you answer the quiz question. For this chapter, you have to answer all the quiz questions and pass up on next week, Monday. So that will be your homework for this week. Okay, so now we're going to look at some of the, we do partial question first. Okay, we do partial question and then we see the video, okay? Okay, question 35. What is the difference between B and T lymphocytes in the immune system? So I open to the floor. Okay, open to the floor. You type your answers once you get your answers, okay? So what will be the answer? Xin Yin Teng say is it C. What about the rest of you? Is the answer C? Yes, answer is C. All of you type C. So let's see whether it's correct. So B cells form plasma cells, correct, which can secrete antibody, correct, into the bloodstream, correct. Okay, it can be secreting into bloodstream or tissue fluid or lymph, depending on where is this plasma cell. All right, and then the T cells, they do not form plasma cells, so correct. Only B cells can form plasma cells. So answer is C, correct. So next question. So the diagram shows part of the immune response. So we've got all these cells, chemical P, cell Q, cell R. So what are PQR? So open to the floor. What is PQR? So is B correct? So let's see. P, pathogen with chemical P on its surface. So P is definitely antigen, right? So these two are possibly correct. Then after that, we got cell Q. P becomes cell Q. So cell Q, is it? So we've got these two choices, T helper cell or B lymphocytes. So usually it should be T helper cell first. Okay, selected and then undergo clonal expansion. So after clonal expansion, so cell Q will undergo clonal selection and then followed by clonal expansion. After that, you will secrete cytokine, right? So cytokine, you got cell R with protein on the surface that can fit to P. So the cytokine will cause the cell R to multiply, right? Okay, so the selected cell R, which can already bind to the P. So this is clonal selection of cell R being activated by cytokine to undergo clonal expansion. That means divide and differentiate into two types of cell. Okay, so active cell is the plasma cell. And then, of course, the second type of cell is the memory cell. Of course, there must be a lot of cells, right? Cells. Okay, plasma cells and memory cells. Okay, so let's continue. So let's see some video.